Okay, well, good morning and welcome. My name's David Whiteman. I'm the Queensland Chair of the Australian Academy of Health and Medical Sciences, and I'm delighted to have with me as my co-convener, in fact, the person who's done most of the hard work, leading a team of people who've done hard work, Isaac Carmichael. So we want to both welcome you to today's Life as a Clinician-Scientist event. Before we go any further, I would like Isaac to acknowledge country. Thank you.
So I'd like to respectfully acknowledge the traditional owners of the land on which we live and work, the Jagera and Turrbal people. I acknowledge the traditional custodians of country throughout Australia and recognise their continuing connection to land, waters and community. I pay my respects to them and their cultures and the elders past, present and emerging, and extend that respect to the Aboriginal and Torres Strait Islander people who may be joining us today.
Thank you, Isaac. I'll just grab that. So I want to particularly welcome those of you who've made the effort to be here today in the audience. It's a gloomy Brisbane day. The humidity is about 100% and we can certainly feel it in the auditorium. For those of you watching online, welcome as well. It's going to be a very interactive event today, so we welcome your participation. So before we go further, there are a few housekeeping announcements. We're still in the midst of a COVID pandemic, and as a health and medical academy, we obviously take this extremely seriously. So for those of you in the room, we're asked actually to keep one seat between each person. So if you haven't got one seat between each person, could I ask you to do that, please? We still have mindful of infection control. And when we're entering and exiting the auditorium, if we could also stay 1.5 metres apart. As I said, today wouldn't have happened without the efforts of lots and lots of people. I particularly want to thank the folks at the Academy who've done the work behind the scenes, but also to an extremely talented group of medical students and PhD students. And I'm going to call them out. They may be embarrassed, but I want to thank Rashan, Catherine, Misa, Chloe, Chang, Ryan, and Elisa, and you will meet them during the day. But those people, we all know how much work it takes to put on a day like today. So all of you, thank you very, very much. A few of our speakers today will be not here in person. They'll actually be presenting online, but we'll make the best with our technology to make sure all of that happens. I won't take up too much of our speakers' time, but I do want to introduce you to the Academy. For those of you embarking on your careers in medical practice and medical research, you may consider in the future interacting a lot more with the Academy of Health and Medical Sciences. So the Academy is one of Australia's learned academies. These are based really on the original academies of ancient Greece, but probably in the English-speaking world, the Royal Society would be the best known of the academies in the modern era. Our academy is a young academy established only 7 or 8 years ago through the visionary work of Ian Fraser, who, as you know, discovered, was behind the HPV vaccine. But the Academy exists to be an impartial and authoritative voice for health and medical science. And you can see in a time like a pandemic, when there's lots of misinformation, lots of confusion, it's very helpful to have people who are not government. They're not representing any stakeholder group. We're not commercial. This is really about the science. And we and others are working in that space. Currently, we have 425 fellows across Australia representing broad disciplines. That's science and medicine, but also allied health and various other craft groups. All of those people are welcome to join our academy as long as your work is in the space of making people healthier and better. And that's what distinguishes us from other academies. Ours is very applied in that sense. And we do a lot of work with other academies and with other sectors in the Australian population to make sure that our advice is heard and that it's reliable and evidence-based. So as I said, our purpose is simple. It's to advance health and medical research in Australia. We want to translate that knowledge so that it benefits all. We foster leadership, so we have mentoring programs for young researchers, young medical scientists in our profession. We try and provide expert advice to decision makers. And we're doing that to make a better Australia that's healthy and happy. So as I say, we do a lot of things behind the scenes, and that's where we have a fantastic secretariat to help us. A lot of clinicians and researchers are busy people, and they just can't possibly get through all of the work that needs to be done. So we do a lot of work behind the scenes drafting policy. And for those of you who are new to this world, I can tell you that an awful lot goes into thinking about the wording that you put into a vaccination policy or into a infection control policy, and trying to ensure you've got the very best evidence so that you're making the best decisions possible. All of that goes into policy making, and it's a very time-consuming but incredibly important role. And it comes out of research. You can't do it without having a research base there. The Academy celebrates excellence. We reward and celebrate the achievements of our finest and brightest minds in the country. We provide a platform for nurturing talented researchers and future researchers, and that's you people online and in the audience today. That's what we're trying to do, is just spark that creative spirit, that spirit of inquiry that will drive you for the rest of your life. And believe me, it is very addictive. And we also support Indigenous health and well-being. And as you all know, there is still a lot of work to do for our Aboriginal Australians. So that's a continuing work for the Academy. Just a couple of quick examples of things that have been happening in the background, and you can download these documents. They're easily available on the Academy website. The COVID-19 advice that's been provided throughout the pandemic from the Academy has been absolutely first rate. It's independent, but it's been ahead of the curve. And many of the things that have come to pass were actually originally put on paper by the Academy's COVID-19 working group. And there've been lots of webinars and things in that space. Research doesn't happen without funding. Funding is absolutely critical. So every year the Academy gets in front of government and makes the case as to why it's so critical to have facilities like this, to have laboratories like the ones here at TRI and at QIMR Berghofer, to provide fellowship and research funding for people who want to do research. It's critically important. If you take that money away, you don't get COVID vaccines, you don't get cancer cures, you don't get dementia care. So you absolutely have to have a research pipeline, and that's what we fight for. And then we have these sorts of events, Life as a Clinician-Scientist. Today you're going to hear from a stellar lineup of busy people who want to tell you how they've managed to balance a life in the clinic with a life on the bench. And as I say, we provide mentorship for people as well. Now, this is beyond me. I'm going to have to ask Isaac what even this means, but it's to do with staying in touch online. Isaac, Can I ask you to take over?
So just throughout today, if you have any questions for the clinicians who are presenting, particularly after the panel discussion or similar, we're recording all questions through Slido. So if you go into sli.do and type in the code J600, that'll connect you to basically our question bank, and we're going to be recording and asking those questions for you.
So I'm wrapping up now. I know you're very keen to hear our speakers today. If you want to keep in touch with the Academy, please visit our website. There's lots of material there, including about our mentoring program, and you can find us on various social media channels. So now I'm going to hand over to Isaac to introduce the introducer for our first session. So Isaac.
All right, so the first volunteer you're going to be meeting today is actually going to be Ryan, who's going to be introducing the first set of speakers for this morning. All right, thank you, Ryan.
Morning, everyone. Please allow me to introduce our first speaker today, Professor Claire Wainwright. Professor Wainwright is a pediatric respiratory physician and co-lead for cystic fibrosis services at the Queensland Children's Hospital in Brisbane, which manages around 450 children with cystic fibrosis across Queensland and northern New South Wales. She is a professor of pediatrics and child health at the University of Queensland, and her research interests include clinical trials, development of lung disease and cystic fibrosis, airway microbiology, and patient-reported outcomes in cystic fibrosis. Today, she will be presenting from genetics to the pharmaceutical market for cystic fibrosis therapies. Please give a warm welcome to Professor Claire Wainwright.
This one here.
Well, first of all, thank you very much for inviting me today. What an exciting opportunity. I wasn't quite sure how to give this talk really, but one of the things that we all of us have in clinical research is a massive challenge. And I quite like hiking. And I actually did manage to get up this to the top here. It's one of the wonderful walks in Victoria. And I think all of us in clinical research have issues with trying to climb some of these cliffs. Oh no, I don't want to look at myself. That's terrible. Really, I don't. Thank you. Okay, all right. So I do have some disclosures. I don't think they're particularly relevant to today, but I do have some. David Reed is an adult physician at the Prince Charles Hospital, and he last year said, I think to the press, we may be the last cohort of doctors to see classic cystic fibrosis. And I think he's right. And it's really, there's been a dramatic change in the outcomes for patients with this condition. Now, when I started working in cystic fibrosis, I was a junior doctor back in the 80s, and patients really didn't survive that long. So on average, patients really died in their teenage years or in early childhood. And we saw what was then known as classical cystic fibrosis. There were already some improvements, but quite honestly, it was not the world of today where we see patients doing extremely well getting into adult life. And I'm going to talk a little bit about that journey. CF is a complex multi-system disorder. It predominantly, people die of their lung disease. So it's chest doctors who look after them, but we tend to have to be fairly general, and we have to work with a lot of colleagues. There's a lot of collaboration because we have to manage all those other systems that are involved. The worldwide burden for cystic fibrosis is very difficult to really understand because in many places, we actually haven't got a clue how much cystic fibrosis is there. But we know that the sort of gene frequency in the population is really quite variable. And this is predominantly a Caucasian disease, but it occurs right across Asian populations and in Africa as well. This was the big discovery. 1989, and it was really in a way the end of a massive effort across international groups. And when you look at it, people were standing on the shoulders of others to get to this point. And in fact, when this paper came out, there were some very disappointed groups, and there were obviously the people who worked together to get this marvelous result were very excited. Now, I'm going to tell you a story of good things and bad things. Now, CF is also how I met my husband. So there are some good things that come out of all of this. Now, my husband had been sent as a postdoc to London to lead the team who were meant to be finding the gene for cystic fibrosis. You can see his name isn't on that, but his work and the work of the group there had really helped to define where the CF gene was on chromosome 7. I was a medical student at the time at Guy's Hospital in London. He had come from Australia. He'd been working at the University of Adelaide. He was an Australian. And I had been at Oxford prior to starting medicine, reading physics. I discovered I really should do medicine and moved to Guy's. And one of my colleagues from physics had moved to London, and she was working at St. Mary's. And when Brandon, my husband, was in London trying to find the gene for cystic fibrosis, they needed patients. They needed a lot of patients. And what they needed were families with different generations to look for this gene. And Lizzie, my friend, who was sharing a flat with him, said, you know, if you want somebody who'll take blood for you, one of the medical students I was at university with might be able to help. And there was a big family in France with 3 generations that all had cystic fibrosis. And I'm half French, I speak fluent French. So she said, I'll ask my friend Claire if she'd do this for you. So it was a blind date. And off we went to collect blood from a family in France, and the rest was history, basically. So that was my introduction to cystic fibrosis as a medical student. It's not really how I got into the research part of it. So let's get back to the science. So it's an autosomal recessive condition. It's on chromosome 7. There are around 2,000 or more variants in CFTR that have been identified, but so far only around 360 have been actually shown to cause cystic fibrosis, and there are some that are really unclear. CFTR is a protein kinase A activated ATP gated anion channel, and it regulates the transport of electrolytes like chloride and bicarbonate across different epithelial surfaces, with really a result of changing the flow of water. Now, the life of a normal CFTR protein is important to really get a feel for, because all of these CFTR variants have problems across all these different areas in this life of a normal CFTR protein channel. And we have to understand where all of those problems are so that we can target the right therapies. And this is what I'm talking about. There are different classes of CFTR variants. Effectively, classes 1, 2, and 3 are the severe end of it, with almost no function or protein available. So let's have a bit of a look at the function of a normal CFTR channel. Well, it has to open. And the open probability is interesting because we always think of it as needing to open all the time, but actually only opens in a normal, healthy cell 40% of the time. It also has to conduct chloride and has to do that effectively. So what determines CFTR activity, quantity, and function? Well, the quantity is quite clearly, let's see if I can manage to do this. Of course, online won't necessarily see this, but quantity is all about the number of CFTR channels at the apical surface, but function is all about the channel open probability or channel gating and the transport rate or conductance. So it's the total that leads to your total CFTR activity, and that's what's very important. And there are different variants that result in either little to no total CFTR activity because of no quantity, so not enough protein, or no function, or a combination of both. Let's look at the most common variant, the F508del. This has almost no protein at the apical surface, but it also, even if you can rescue the F508del protein and get it there, it's got a problem with channel gating. And so that's a sort of double whammy. What about the G551D? That's another variant where you get, in fact, lots of protein, but it's like having a locked door. It doesn't work. And of course, that has a different approach. Now, how do we diagnose CF? Well, by heel prick here in Australia. And I'm not going to go through the details of that because I haven't got time, but effectively the actual diagnosis is made genetically and through sweat testing because CFTR affects your sweat gland. So people with CF will have higher sweat chlorides. They'll produce sweat that has more salt in it. And this is really the gold standard for making the diagnosis. And what's sort of important is this level of 60. And I do want you to remember that 60. There is a relationship between genotype, phenotype, and CFTR function. And this is all a bit small to read, but effectively, if you've got no CFTR protein, you're going to have a severe phenotype. And somewhere in between, there's some middle ground. If you're a carrier, then you obviously don't have the disease either because it is an autosomal recessive. Airway infection is incredibly important in this disease. And you can see an end-stage lung on this side of the, on the right side of the slide. And that's what it looks like when you pull it out. And it's full of pus. Again, this is just a slide pulled out of the US data registry showing that infection happens early and it goes throughout life. And by the time you're an adult, most patients have a chronic infection, usually with Pseudomonas. Now, the clinical phenotype is also influenced by other factors, not just your genes. It's also all the other genes that you have along with the CFTR gene. And the modifier genes do impact, we do know that, but it's also the environment. So it's your environment early on, how many viruses you get, whether your parents smoke, all sorts of things impact. But what's happened since the gene was discovered? Well, what I'd like you to look at is the green here, and that's normal lung function. And from 1989 to 2019, you can see that in 18-year-olds, we now have nearly half of them have normal breathing test results. So that's a big difference from that tiny slice of the green pie. Now, the red dots are going, their sort of lung function against nutritional outcomes. And again, you can see almost a straight line of improvement with time. Again, the green, if you look at that here, is related to some of the good things in adults. And what I want you to really get a feel for here is that 3/4 of people, adults with cystic fibrosis, are either students or working. So that's pretty good as well in terms of quality of life. Now, improvements in outcomes have occurred for all sorts of reasons: specialist multidisciplinary care, newborn screening, improved nutritional status. I've highlighted in red Pseudomonas eradication and management of chronic infections, because that has had a dramatic effect and really changed the story. The prevention of pulmonary exacerbations goes along with that, and new therapies. And I'm going to talk to you a little bit about those red items, because that's where I've been focused through my life. This is the survival curve over time. And this comes from the 2019 US CF Foundation Patient Registry, which is the biggest registry in the world for cystic fibrosis. So it's probably the most robust in terms of looking at epidemiological data. But our data here in Australia look very similar. Those blue lines refer to 2 events, really. The event at the bottom, so let's see if I can make this work. So here, what you're seeing is where we first had inhaled treatments for Pseudomonas infection. You can see there's an increase in that graph in survival. And this is where CFTR modulation started. So those are the drugs that have impacted the actual basic defect. But you can see that there were improvements all the way along, all the way through. Now I'm going to tell you a story about a fairly amazing guy called Bruce Montgomery. He was working for Genentech and was involved in developing human recombinant DNAs, but also actually, interestingly, for those who know about Xolair, for the basics against which Xolair was discovered. Now, he recognized that we needed some inhaled antimicrobials. Prior to this, physicians were using the version that we give IV as a nebulized form. And that's what physicians do. Clinicians use what they can. And we knew that we needed to treat these terrible infections. You saw those lungs, we needed to treat them. So people were given IV antibiotics, but between those IV antibiotics, people were getting sick. What do? Well, you give them these inhaled therapies. But of course, those medicines are not made for a nebulizer. They're not made to be inhaled. So Bruce worked to try and develop this, and he developed the first inhaled antimicrobials, along with the nebulizer, because of course, how you inhale these things is critical. So he worked with the engineers in Pari to develop a kind of combined approach. And in fact, he mortgaged his house to do this. He set up pathogenesis, and he worked with clinicians. FDA approved the drug in 1998, and it was PBS listed here in 2011. You'll notice there was a bit of a gap with that. And some of us got fed up with that gap and in fact worked with PBS to try and get that approved. So this is part of my story as well. So I completed my fellowship in Melbourne in 1997. I returned to Brisbane as a respiratory consultant. Now, I got my first NHMRC project grant as a CIA in 1999. I had absolutely no track record, absolutely zippity-doo-dah. I'd barely published a paper. I'd never got a grant. I'd never done anything, but I had a good idea. And I also was lucky enough to be trained as far as statistics and clinical trials by John Carlin in Melbourne, who ran the epidemiology side of things and the research statistics programs. And I thought that we needed to, when I looked around at my patients, I recognized that they were infected. Young children don't cough up mucus very efficiently. How do you know whether they're infected? So people were starting to do bronchoalveolar lavages. And when I went to conferences, I was finding that people were doing hundreds of these. Were they useful? Was it going to change what we did? Well, we set up the trial to do it. And I had a wonderful collaboration with different physicians around the country. Now, I was invited to a meeting in Sydney by another amazing physician, Dr. Peter Cooper, who was a respiratory physician at Westmead. And he was particularly involved in setting up the CF data registry, which has been hugely important in Australia. And he had invited Bruce Montgomery to present at that meeting. Well, I was very junior. I didn't really want to ask any questions, but I found myself at the end of the meeting in the lift with Bruce Montgomery. And the lift got a bit stuck and we were sort of standing, staring at each other. And I thought, well, you know something, I want this drug and I want this drug for my trial, because if you're going to try and treat Pseudomonas, then you want the best treatment. So I asked him in the lift, would you help me? Would you give us this drug for free? And he looked at me and said, sure. And we kept up a good friendship, and we ended up using his drug. That led to all sorts of changes. It led to an invitation to give a plenary at the North American CF conference in 2009, because by that stage, I had been involved in a number of research programs with the US teams through the CF Foundation. It led to my first big paper in cystic fibrosis, which was this trial, which was published in JAMA. And it led to discussions between Peter Cooper and Bruce Montgomery over drinks, I might add, about the use of another drug, which had been used in its IV form and that smelt disgusting in its IV form and was being used as inhaled therapy at Westmead for end-stage lung disease. This was called aztreonam. And Bruce took that conversation and then developed aztreonam lysine Caistin, again with a special nebulizer. And I worked with Peter Cooper and with Bruce to again do those trials. So that was an exciting outcome. So I'm now going to switch a little bit. So by this stage, I had been involved with some groups in the US, and I was, I suppose, perhaps starting to get a bit more well known, which helped with the start of my work in CFTR modulation. Now, again, the story of CFTR modulation is one of amazing people. So you will all have heard, I'm sure, of Roger Channe, who discovered along with other people, of course, the green fluorescent protein, and he received the Nobel Prize with 2 others in 2008 for that discovery. Now, he also set up Aurora Biosciences in San Diego, and that was a company that was looking at high throughput screening. Now, Bob Bell was the first CEO of the CF Foundation in the United States, and he was an extraordinary man. He worked with another Bob, actually, who was a parent, and they had a new approach to how to do this. They moved to Washington, they lobbied like crazy and got lots and lots of money. And they also prioritized research, and they wanted to find a cure for cystic fibrosis. So Bob did some odd things that really have paid off dramatically. So he said to Aurora Biosciences, we will give you money for you to concentrate and find drugs that we need. And they basically invested $40 million in Aurora. And Aurora then got bought by Vertex Pharmaceuticals. And again, this has led to a historical change in the types of treatments. The first was a potentiator, ivacaftor, which had dramatic outcomes. So if you remember that sweat chloride I told you about was the 60, the magic 60, these children given ivacaftor had sweat chlorides that were below this red line, these lines which give you the 60 mark. So it dropped below that. So for the first time ever, they had functional CFTR. Now, this was an easier variant to work on because you've got protein there. So the potentiator opens the gate, basically. And so this was the first successful outcome. And we've looked at it down the track, and even in the clinic, not in trials, it has had dramatic effects on reducing death, reducing organ transplantation, and improving outcomes. The sort of, if you like, story of it over time, 2012 was when it was registered by FDA and EMA. And now in 2020, eligible infants from 4 months are being able to use that in the US. Here, it's funded for children with these specific gating variants from 12 months, and that's had a dramatic effect on outcomes. But of course, that's only around 4% of the population. So what are we going to do about all the rest? Well, the most common, of course, is the F508del. Now, this is tricky because you have to not just rescue the protein, but you also have to open it up. So with ivacaftor alone, you get almost nothing in terms of effect on the F508del protein. Lumacaftor alone, not enough. But if you give both, you get sort of a much better signal. And so that led to a trial, a very large trial, that one of the biggest trials at that time, over 1,000 patients worldwide with cystic fibrosis. And these patients were homozygous, had 2 copies of the F508del protein. That was a modest improvement that was found. So it was clearly not enough, but it was the signal that we knew that this type of approach was going to work. And this led to other studies, including for younger patients, which has sort of come to now PBS listing in 2019 for 2 to 5-year-olds who have access to this drug. Further drugs have been developed, including Symdeko, which is a tezacaftor-ivacaftor combination. And one of the problems with the Orkambi, the lumacaftor-ivacaftor, was that the 2 drugs interacted. So these are tricky things. And when you go around the original laboratories in San Diego to look at where all of this happened, it's quite interesting. You see where they've had explosions of things in their lab. You can see where they've had sort of these amazing findings. And one of the things that they really struggled with was the dosing for Orkambi, because the lumacaftor and ivacaftor actually interact in terms of liver metabolism. So since the drug was never going to be the complete winner for people who are homozygous the F508del protein, how are we going to add in another drug to make it even better? So they needed something that was stable. So then they went to one of the other correctors, the tezacaftor, and the tezacaftor then got added to the ivacaftor. And this then led to a whole range of new studies, again, showed very similar results to the Orkambi studies, but this formed the basis or the backbone for adding another drug. And so this is where the phase 3 trials of the triple therapy, which is now sort of hitting the headlines, has come. And again, massive impact on sweat chloride. And this is not just in patients who are homozygous or have 2 copies, but also in patients who have 1 copy of the F508del. So that now takes us to almost 3/4 of the CF population. So that's pretty exciting. There's still a long way to go and patients still have disease, but at least we're on that way. So this is the sort of total story, if you like, across from finding the gene through to sort of the last few years. And there are now multiple different companies and approaches to different parts of the CFTR protein defects with corresponding therapeutic strategies. So what have I learned through all of this? Well, it's the bench to bedside story for me has been all around collaboration. We've collaborated with pharma at different levels. We've helped them to design trials. We've helped them to develop outcomes. We've helped them to, if you like, look at their results in a way that is useful for clinicians. There's been a lot of luck on the way. There's been a lot of persistence. It's also collaboration with patients. It's collaboration with government. It's working with PBS. It's working with government for funding. But all of it is standing on the shoulders of others, actually. And all of us, the little bit that we add is always on the top of all the other things that other people have done. And so it's trying to sort of find, if you like, your role in that puzzle and enjoying that journey. So one of the things I wanted to also say is that our patients play an enormous part in all of this. Kari Lee Venning was one of my patients. She unfortunately died of cystic fibrosis, but she was an amazing photographer, and this is taken on the Sunshine Coast where she lived. There are also amazing people who've helped me on my journey. And as a clinician-scientist, I suppose, one of the things that I always found was that you're never quite in the right for anybody. The scientists think you're playing at science and that you've already got a real job, so it's not that important for you. The clinicians think that you're shirking because you're not doing your clinics enough. So basically, you're always in trouble as a clinician scientist. But actually, there are all sorts of fantastic mentors out there who've kept me at it, and some wonderful people who've given me jobs and supported me. So my huge thanks to all of those. So I'll end there.
Are there any questions?
I'll start. Is that working? Is that working? Yeah, that's working. So maybe I'll start with one while people type away and other people think up one. So I guess, how do you deal with, I guess, that comment you made about always being in trouble, about, I guess, juggling the expectations of the people around you? I mean, I guess having mentors and people who can, I guess, work top down is incredibly helpful, but how do you manage that yourself personally?
I think you need to hide like a rhinoceros. You know, I remember sending my first paper in and it got bounced so fast and I kind of went, huh. But I also, I suppose it's called just trying to keep going. You have to have a focus. And my focus, I suppose, is going to the clinic and seeing patients. And when you see your patients and you chat to patients, you find out what they need and you're actually their advocate. And as a medical student, I remember being taught that the patient in front of you is yours, no matter where you are in the hierarchy, and that you are their advocate and you need to fight for them. And that's actually incredibly important. And so you do whatever you can, either as the clinician in front of them or as the researcher or as both. And I think that's incredibly important. The other side of it is that there's an ethical balance sometimes between getting called for things and being asked to do things. So you have to also, I think, be careful, because working with pharma has its dangers. And people have often said to me, well, how do you work with pharma? And are you a dirty player? And kind of have to explain to them that if you don't work with pharma, you never get any drugs. So it's kind of important, but I never take money from pharma, and I am very, very careful about that. So it's balancing those things as well, and balancing your family, because actually, if you have a family, they are the ones who keep you sane. And I've been super lucky because I have a very, very tolerant family. Who have been amazing and have not minded my doing my grant writing at 1 o'clock in the morning and various other things.
Yeah, okay. Yeah, that was just a wonderful presentation. So thank you for sharing that. You mentioned in your talk that your first research grant was obtained without you having any real training at all in research. What advice would you give to a new generation of people thinking about research? Should they get some training in research before attempting to write grants and compete in that space, or just give it a go?
Well, I mean, I'd had no, it's not that I'd had no training. I'd done, I got an MD and I was close to submission by the time in fact, I had submitted and I got my MD by the time I got my grant. So I suppose I'd had some training in research. I think you have to just give it a go. These days, I don't think it's possible to do what I did. I think if you put in a grant to an HMRC like I did, you wouldn't get anywhere. In those days, people took chances. And this was the first ever intervention trial in cystic fibrosis across a multi-center group. And it was the first one in newborn screening patients. So I think there was some excitement around that. And I also had I didn't go into it alone. I went to in those days we had interviews. to get the grant. And I went in with my whole, panel. I had John Carlin with me, who was a senior statistician. I had, senior physicians with me who could back me up and say that they were going to do this with me. So I kind of looked vaguely credible as a result of that. And we did a lot of practice. So they took me through it. They were a bit shocked that I'd got to the stage of having an interview, I think. And we did practice after practice after practice until, I was, fed up with it. Where they were throwing questions at me. And in the end, I felt quite confident going in. So I guess I don't know these days how people negotiate that first grant. I mean, I got my first paper really by having huge help from an amazing editor at the New England Journal. My first research paper was a New England Journal first author paper, and that was with no money at all. And that was a trial that I set up because one of my colleagues had asked me about the use of epinephrine. And I went, well, I've no idea. And we had a go at doing it. And again, it was regional pediatricians and our hospital. We worked together, we did it, we had no money. And I wrote the paper, sent it off to the New England Journal thinking, well, that was a paper, that was a journal I'd heard about. And I got back and the editor who some of you may know, Jeff Drazen, it was in the days of Old Post, had basically red marked everything and had given, big queries all over the place. I had to rewrite it. I had a fabulous statistician working with me. And in the end, it got through. Now, it was just extraordinary that I was lucky. I was super lucky. And it was also daring. I think you try, you give it a go. I mean, what's the worst? The worst is they say no. Who cares? If they say no, you move on, you've lost time. But, you give it a go. So I think that's the only thing I can say. Give it a go. If you've got a good idea, go for it. Believe in it, work on it.
Other? Okay, so is that?